Hello students, wish you all a very happy and prosperous new year. Welcome again to the channel Physiology Redefined. In my last video, I tried to give you the basic idea regarding the neuromuscular transmission. So there I told that if a motor neuron is stimulated, then the muscle membrane is first excited or stimulated. And this is the process in which the stimulation of the motor neuron leads the excitations of the muscle membrane, that is the sarcolemma, and that process is known as the neuromuscular transmission, which I already talked about it. Now, as I told that if a motor neuron is stimulated, then muscle membrane or sarcolemma is excited or stimulated, and that is the first step of the response given by the muscle, that is in the form of muscle contraction. So first step of the muscle contraction is the excitations of the muscle membrane in response to the stimulation of the motor neuron. Now in this video I will explain if a motor neuron is stimulated, then first the muscle membrane or sarcolemma is excited or stimulated, and in response to stimulation of this muscle membrane or sarcolemma, the contractions of the muscle takes place. And in this case, what's happened? First, there is a excitations of the muscle membrane, which is known as the electrical events, electrical phenomena, and then the muscle contraction starts, that is the mechanical phenomena. So, electrical phenomena ultimately leads the mechanical phenomena. So two individual phenomena, two individual events are linked by a mechanism which is known as excitation contraction coupling. So today I will talk about the excitation contraction coupling. So excitation contraction coupling means, so as per the definition is concerned, that is excitation of the muscle membrane, excitations of the muscle membrane, this leads to the contraction of the muscle, contraction of the muscle. So this is the phenomena which is known as the electrical phenomena, and this is the phenomena which is known as the mechanical phenomena or electrical event and the mechanical event. And this mechanical event to take place, the electrical event is necessary, and these two event, electrical event and the mechanical event, is joined, is linked by the help of a coupler, that is the linking or linker, coupler or linker, and this linker or coupler is the calcium ion. So this calcium ion is the linker or the coupler. That means this calcium ion, or by the help of the calcium ion, electrical phenomena gets coupled, gets linked with the mechanical phenomena of the muscle. So if a muscle is stimulated, then mechanical events of the muscle, that is contraction, is initiated, and that initiation is coupled, is linked with the excitations of the muscle membrane by a coupling agent known as the calcium ion, and thereby this entire phenomena, that is excitations of the muscle membrane that leads the contractions of the muscle by the help of a coupling agent known as calcium ion, is known as excitation contraction coupling. Excitation contraction coupling. Two events, electrical events is linked with the mechanical events, and by the help of a linker, that is a calcium ion, or coupling agent, that is known as calcium ion. So this phenomena is known as excitation contraction coupling. So in this video I will talk about the details of this excitation contraction coupling. So to get a clear idea about this phenomena, please watch this video till the end. Now, to research the mechanical events in response to the stimulation of the muscle membrane, a specialized structure is held responsible. That means the components that is responsible for the excitation contraction coupling. Components responsible for excitation contraction coupling. What is the component? That is the sarcotubular system. Sarcotubular system. So what is this sarcotubular system? Sarcotubular system, this is a specialized system present in the skeletal muscle, present in the muscle, which is comprised of basically two components. First one is the sarcolemma, or the cell membrane or the muscle membrane. So this is the sarcolemma. This is the sarcolemma. This is the first component. So this has got two components. One is the sarcolemma. Now the sarcolemma, or the muscle membrane, invaginates towards the myofilaments. They go deep towards the myofilaments. So here is the myofilament, muscle contractile element, myofilament. So this is the contractile elements of the muscle which is responsible for contractions of the muscle. Now, this sarcolemma, muscle membrane, goes deep or invaginates towards the myofilament in a tubular structure, and this is known as the T-tubule. So this is the structure which is known as the transverse tubule or T-tubule. So first component of the sarcotubular system is the T-tubule. So T-tubule is the inward bulging of sarcolemma, inward bulging of the sarcolemma, or inward invagination of the sarcolemma, that is the T-tubule or transverse tubule. Second component of the sarcotubular system is actually the tubular network of the endoplasmic reticulum. So this is the endoplasmic extension of the endoplasmic reticulum, a long tubular network of the endoplasmic reticulum. So this is the endoplasmic reticulum. The endoplasmic reticulum's long extended tubule is known as the L tubule. So this is the structure which is known as the L tubule or longitudinal tubule. So second component is known as L tubule. So this is the part or extended part of sarcoplasmic reticulum. Part of the sarcoplasmic reticulum. And this part of the sarcoplasmic reticulum that is known as L tubule, this is extended, this is ended as a solent structure, this is ended as a solent structure, and this solent structure is known as terminal cisternae. Terminal cisternae. So, L tubule is the tubular part of the sarcoplasmic reticulum, and this ends as a solent structure, which is known as the terminal cisternae. Terminal cisternae. And this terminal cisternae, this contains or stores the calcium ions. The numerous calcium ions, plenty of the calcium ions are stored within the terminal cisternae. So numerous calcium ions are stored within the terminal cisternae. So terminal cisternae contains or stores calcium ion. And here you see this T-tubule invaginates towards the myofilaments, and this actually invaginates towards the myofilaments at the AI junction. This invaginates at the AI junction. This invaginates at the AI junction. What is AI junction? That I will discuss in another video. So this invaginates at the AI junction, and in a sarcomere, that is a structural unit of the muscle. Details of these things I will again discuss in another video. So in the sarcomere, in case of skeletal muscle systems, these one T-tubule is associated or is closely attached with two terminal cisternae. Here you see one terminal cisternae, another terminal cisternae. Number 1 terminal cisternae and number 2 terminal cisternae. And this is the structures in which a T-tubule is attached or closely connected with two terminal cisternae. One T-tubule is functionally linked with two terminal cisternae, and this structure is known as triad. So, in the sarcotubular systems, what's happened? Two terminal cisternae plus one T-tubule. One T-tubule in skeletal muscle. This is known as triad. Whereas in case of cardiac muscles, what's happened? one terminal cisternae plus one T-tubule in cardiac muscle. This is known as dyad. So, in case of cardiac muscle, structure is known as dyad because one T-tubule is functionally connected with the one terminal cisternae. But in case of skeletal muscle, one T-tubule gets connected with two terminal cisternae. So, in a sarcomere, two terminal cisternaes are there, which is closely associated with a one T-tubules, and this structure is known as the triad in case of skeletal muscle and dyad in case of cardiac muscle. So this is about the components of the sarcotubular system, which is responsible for releasing calcium ions, stored calcium ions, and that calcium ion is ultimately responsible for initiating contractions of the myofilaments or the muscle once the skeletal muscle or the muscle is getting stimulated. So let us see the mechanism of excitation contraction coupling. So now is about the mechanism of the excitation contraction coupling. Mechanism of excitation contraction coupling. So, as I told that, if a muscle membrane is stimulated, suppose this muscle membrane is stimulated. So, stimulated by the motor neuron. Muscle membrane is supplied by the motor neuron, and this motor neuron releases the acetylcholine at the neuromuscular junction. Now this acetylcholine gets bind with the ligand-gated receptors, ion channels present on the muscle membrane, and thereby there is an entry of the sodium ion, and this sodium ion entry ultimately leads the initiation of the action potential of the sarcolemma. So initiation of the action potential means there is a depolarization of the sarcolemma. So inside will be now electropositive in respect to outside of the membrane. So in the resting condition, inside was electronegative. Once the muscle membrane is stimulated, then muscle membrane gets depolarized or excitations of the muscle take place. So here we can say that muscle inside of the membrane, inside of the sarcolemma will be electropositive. Now this electropositive means this electropositivity will be spreaded across the cell membrane towards the myofilaments. Here, as I told earlier, here is the myofilaments. So inside will be electropositive and outside will be electronegative. Inside will be electropositive, outside will be electronegative. This is the status of the T-tubular membrane. That means once the motor neuron is stimulated, so if the motor neuron is stimulated, or once the motor neuron is stimulated, what's happened? As I told, there is a release of acetylcholine at neuromuscular junction, and this results sodium entry to inside of sarcolemma, and thereby there is a excitation of or depolarization or excitation of sarcolemma. So sarcolemma is excited or depolarized. Once the sarcolemma is excited or depolarized, now there is a inward spread of action potential through T-tubule. Through this T-tubule, action potential is passed towards the myofilaments. So inward passage of the impulse through the T-tubule by the help towards the myofilaments through the T-tubule. So this is the T-tubule. So inward passage of the impulse through the T-tubule towards the myofilaments. Now what's happened? Here you see again. On the T-tubular membrane, there are some receptors which are present on the T-tubular membrane. So T-tubular membrane, some receptors are there which are known as dihydropyridine receptors. So these are the known as dihydropyridine receptors. Dihydropyridine receptors or DPH. Dihydropyridine receptors. Dihydropyridine receptors. Now, these dihydropyridine receptors, they are basically the voltage sensor. They sense whatever voltage fluctuation takes place at the T-tubular membrane, they detect that change. So dihydropyridine receptors, they act as voltage sensor. So they are the voltage sensor. As I told that the inward passage of the impulse, that is depolarization wave, is passed from outside of the membrane towards the myofilament through the T-tubule, and that means outside is electronegative and inside is electropositive. So some change of the voltage is there, and as this change of the voltage is there, that change of the voltage across the T-tubular membrane is detected by the dihydropyridine receptor. Once this voltage change is detected by the dihydropyridine receptor, they sense the signal to the terminal cisternae. Here I told there is a terminal cisternae which stores the calcium ions. This stores the calcium ions that I already talked about. So this is the terminal cisternae. So on the terminal cisternae, what's happened? There are some proteins on the membrane of the terminal cisternae. Again, there are some proteins which are known as ryanodine receptors. So these are known as ryanodine receptors. And these ryanodine receptors, are basically the ligand-gated calcium channels. So these are basically the ligand-gated calcium channels. So what's happened? Once the dihydropyridine receptor gets aware of the fact that voltage across the tubular membrane gets changed from inside electronegative to inside electropositivity is there, then this dihydropyridine receptor send the signal to the ryanodine receptor, and this results the release of the calcium ions. So calcium ions will be released. Through these calcium ions from the terminal cisternae are released through these calcium ligand-gated calcium channels or ryanodine receptors. So this ultimately results the release of the calcium ions from the terminal cisternae. So from the terminal cisternae, whatever calcium ions remain stored, they are now released via the ligand-gated calcium channels, which are basically the ryanodine receptor protein. And this is how the calcium ions are released inside the sarcoplasm. And once the calcium ions are released inside the sarcoplasm, they binds with troponin C. They binds with troponin C molecule, and that ultimately is responsible for contractions of the muscle, and the details of these contractions of the muscle I will discuss in another video, where I will discuss about the mechanism of muscle contraction or molecular mechanism of the muscle contraction. So under the excitation contraction coupling, what's happened? Here you see, so inward passage of the impulse, subject to inward spread of the axial potential through the T-tubule. This results the sensing of voltage change across T-tubule by the dihydropyridine receptors. So this dihydropyridine receptor gets aware of the fact that some voltage changes takes place. Once these dihydropyridine receptors are stimulated or excited, then what's happened? This is responsible for passing of the signal. So they pass the signal to ryanodine receptor. Ryanodine receptor present on terminal cisternae. Membrane, ryanodine receptors are there, which are now stimulated, and this results the configurational change of ryanodine receptor, and this configurational change of the ryanodine receptor, that means the opening of ligand gated calcium channels. Ligand gated calcium channels. So release of the calcium ion, release of calcium ion from terminal cisternae. From the terminal cisternae, calcium ions are released into the sarcoplasm, into sarcoplasm. Now what's happened? This calcium ion in turn is responsible for binding of calcium ion with troponin C molecule, and this troponin C molecule binding with calcium ion, this is responsible for configurational change of the troponin molecule, and this ultimately results the uncovering of the actin binding site of the myosin head. Generally what's happened? The muscle contraction take place because of the actin myosin complex formation. And this actin myosin complex is not formed in the resting condition because they are covered, their active binding site is covered. So both cannot come in contact. Myosin head cannot come in contact with the actin molecule because they are covered by troponin and the tropomyosin. The details of these things I will again discuss in another video. Now what's happened? Once calcium ions are released, they bind with the troponin C molecule. So configurational change of the troponin molecule will take place, and that leads the uncovering of the actin binding site of the myosin head. So myosin head is no longer get prevented to bind with the actin molecule, and thereby actin myosin complex is formed. So this is responsible for uncovering of actin binding site of myosin head, and this ultimately responsible for cross bridge formation or actin myosin complex formation, and this is responsible for muscle contraction. So this is how the excitations of the motor neuron results the excitations of the muscle membrane, and that in turn is responsible for contractions of the muscle. And in this case, two phenomena, electrical phenomena gets coupled with the mechanical phenomena by the help of coupling agent calcium ions, and thereby this phenomena is known as excitation contraction coupling. Now this excitation contraction coupling in case of cardiac muscle is little bit different. In case of, this is the explanation that I have given in case of skeletal muscle. In cardiac muscle, the basic mechanism is same with some difference. What are those differences? First difference is that in case of cardiac muscle, what's happened? EC coupling in cardiac muscle, some basic mechanism is same. There are some minor difference. First difference is that in case of cardiac muscles, this dihydropyridine receptor, they act as voltage gated calcium channels. So these dihydropyridine receptor act as voltage gated calcium channels through which calcium ion from extracellular fluid. So from this is the extracellular fluid. From this extracellular fluid, calcium ion enters inside. So through which calcium ion from ECF enters sarcoplasm. This is number one difference. In case of skeletal muscle, no extracellular source of calcium ions are taking place. But in this case, extracellular source of calcium ions are taking place from the ECF to the sarcoplasm. Second difference is that once the calcium ions are entering inside the sarcoplasm, this calcium ion triggers the release of the calcium ion from the terminal cisternae through the ryanodine receptor, which act as ligand gated calcium channels. So in this case, ryanodine receptor acts as ligand gated calcium channels, and thereby what's happened? The calcium ions gets released from terminal cisternae. Stored calcium ions gets released from terminal cisternae through these ligand gated calcium channels, and this mechanism is known as calcium induced calcium release. This, which is very, very important in case of cardiac muscle. In case of skeletal muscle, this calcium induced calcium release is not that much significant. But in case of cardiac muscle, this CICR or calcium induced calcium release is very, very significant. And third difference of the acceleration contraction coupling in cardiac muscle is that in case of cardiac muscles, there are some proteins, there are some calcium binding proteins on the T-tubular membrane. There are some calcium binding proteins on the T-tubular membrane, and these calcium binding proteins are known as sequestrin. So this sequestrin normally binds the calcium ion. So once the action potential is passed through the T-tubular membrane, sequestrin protein no longer binds the calcium ion. So sequestrin protein now release the calcium ion, and that calcium ion again enters inside the sarcoplasm. So in this case, what's happened? On T-tubular membrane, there are sequestrin protein which binds calcium ion normally. So when action potential passes through T-tubule, then what's happened? There is a release of the calcium ion, from sequestrin. And this calcium ion enters enters sarcoplasm, and thereby the plenty of calcium ions are released inside the sarcoplasm. Plenty of calcium ions. First one, so there are triple source. First one is the extracellular fluid via the dihydropyridine receptor or voltage gated calcium channels, L type of calcium channels. Second one is the induced calcium release from the terminal cisternae, and third one is the calcium again is released from the ECF, which gets bound with the sequestrin proteins. So because of these three sources, the plenty of calcium ions are released inside the sarcoplasm, and thereby that is responsible for very vigorous, very powerful contractions of the cardiac muscle. So this is all about the excitation contraction coupling. Hope the concept is clear to you. And if you like this video, please subscribe the channel and don't forget to press the like button. Thank you.